Hi guys, this is Mrs. Holmer, and today we're going to be reviewing biochemistry for the end of course exam. The three topics of biochemistry we'll review are water, enzymes, and macromolecules. Let's get started with water and its properties. So water is a molecule made out of one oxygen and two hydrogen that are covalently bonded together, just meaning that the electrons are being shared between the oxygen and the hydrogen. Now, they don't share evenly, so they share unfairly. And so, because of this, oxygen takes on a slightly negative charge and hydrogen takes on a slightly positive charge. So, we call this a polar molecule, meaning it has opposite side charges. As a result, the hydrogens that are positive can be attracted to other things, forming what we call hydrogen bonds. And hydrogen bonding is what creates all of water's awesome properties. We'll also see hydrogen bonds holding DNA together in the center, and we'll see covalent bonds holding DNA together on the sides. So again, water has covalent bonds holding the molecule together and hydrogen bonds holding the different molecules of water together. So hydrogen bonding creates all sorts of properties for water. First of all, it creates cohesion, which means water molecules will stick to other water molecules, which is why we see water droplets and also why when water spills, it seems to stick together. We also see adhesion, which is when water sticks to another molecule, like glass or a leaf. We will also see something called capillary action. Capillary action just means that water is being drawn up against the force of gravity. And this is how water gets to the top of a tree or the top of a plant against the force of gravity. It's using both cohesion and adhesion to draw it up. All right, the next topic we're going to look at are the macromolecules. And just a reminder that most of life is made out of just a few elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And these elements are going to make up what comprise the four main molecules that make up cells and make up life, which are the macromolecules. So the macromolecules that we'll need to know are carbohydrates, proteins, nucleic acids, and lipids. The first three, proteins, carbohydrates, and nucleic acids, all are considered polymer, which means they're big molecules, but they're made out of smaller repeating pieces. So we'll look at each of these individually. Proteins are also known as polypeptides, and they're made up of amino acids. So there's only 20 amino acids that make up all of proteins. So I remember we say proteins are mean. They're made up of amino acids. And there are only 20 amino acids that make up all of proteins. So the protein's function is going to be determined by the order of these amino acids and how that folds into a particular shape. So enzymes are a protein, and enzymes are very important to most biological processes. Other functions of proteins including building, regulating, fighting disease, controlling cell processes, and communicating between cells. Carbohydrates are the next group of polymers and that make up a cell. Their polymer name are polysaccharides, and they are made up of monosaccharides such as glucose. So remember we talked about photosynthesis, how it makes glucose. Well, now we can take glucose and we can build it into these other carbohydrates, which are building materials for the plants. Carbohydrates are used for building materials as well as for short and long-term energy, which is why you might grab a carb before you do a race. The last set of polymers or macromolecules are nucleic acids. These are DNA and RNA. Nucleic acids are made up of nucleotides, and those nucleotides are built together in two different ways, either a double helix for DNA or a single helix for RNA, which we'll look at in another video. And of course, nucleic acids are used to store and to transmit genetic information. So these are the main macromolecules. There is one more, and that is lipids. Lipids are fats, oils, and waxes, steroids, and cholesterols that store long-term energy, provide insulation and warmth. So there's storage for that glucose. If we have too much, it stores that. It's also waterproof coverings. And hello, we studied how all the membranes, not only in the cell, but in those double membrane organisms are made, or those double membrane organelles are made up of lipids, those phospholipid bilayers. And so lipids are so important to your cell because it's your cell membrane and it controls what goes in and out of the cell. All right, part 3 of the first unit was enzymes. So enzymes are so important. We've seen them all throughout biology. So enzymes are proteins, and they're acting as catalysts, which means they speed up reactions. How they do that is by providing a spot for the reaction to happen and lowering the activation energy. So enzymes are very specific. They have a very specific shape that's obtained by this amino acid sequence being built properly and then folding down into that shape. And they attach to just one particular thing called their substrate. So each enzyme has its own substrate that it works on and its own chemical reaction that it catalyzes, that it speeds up. And so the enzyme here attaches to the substrate, does the reaction, lowering the activation energy, allowing it to speed up, and releases the products. Enzymes, however, throughout the entire process, keep the same shape. The reason they keep the same shape is so they can be reused again and again and again. Eventually, they'll break down, and your body will need to recreate the enzyme through protein synthesis, which we're going to review in another video, to create those enzymes so that we can catalyze or speed up reactions. Now, there are many things that affect the rate of enzyme activity. So enzymes can only work so fast, but certain conditions will allow them to move faster or to go slower. The first thing that affects the rate of enzyme activity is temperature. So cold temperatures are usually not the greatest, and super hot temperatures are also very bad for enzymes. But enzymes will work at a very specific range of temperature, and that temperature is different for every single enzyme. Enzymes in our body will typically have an enzyme temperature graph that looks like this. Cold temperatures, not so good. At body temperature, awesome. And then after we pass that body temperature, it drops sharply. And so you can check the optimum range of temperature for each enzyme by looking at where is the rate of enzyme activity highest. It's that temperature that's best for that enzyme. But remember, every enzyme works better at a different or specific temperature that's unique to it. Not every enzyme will work at the same temperatures. There are some enzymes that work at very high temperatures and some that work at cold temperatures. The next thing that affects enzymes is the pH. So pH is so important to an enzyme because it can change its shape and cause it to not be able to do its job. So pH, each enzyme works better or best at its own specific pH, whether that's an acidic pH below a 7 or a basic pH above a 7, pH will affect how an enzyme works. And so when we've been looking at ecology and looking at how we're altering the world in terms of temperature and pH, that's directly affecting the enzyme activities in different biological processes because temperature and pH affect enzymes, which speed up biological chemical reactions. The last thing I'll touch on with enzymes is that if an enzyme is exposed to high heat or extreme pH, it will completely change the shape and break down my enzyme. The enzymes, when they lose their shape, cannot do their job because they cannot attach to the substrate. And this is called denaturing. Because remember, the shape is what is so specific about an enzyme that if we change the shape, the enzyme can't do its job and it will have to be remade. All right, that's it for biochemistry review: water, macromolecules, and enzymes.